Welcome back, learners, to my virtual lab. Today, in my virtual lab, we will be discussing a new biochemical test, and that is your urease production test. So today in my virtual lab, I will be discussing your urease production test. So for urease production test, let's move back to the history of the urease production test. Now you know that this is basically done by the help of your urea agar. Okay? And this urea agar was developed by Christensen in 1946 for the differentiation of your enteric bacilli. So they basically are used for the differentiation of your enteric bacilli. And the urease test is used to determine the ability of an organism. So it is basically the ability of the organism to split urea. So it is basically the ability of the organism to split urea, and this particular splitting of urea is basically done by the urease enzyme. So urease enzyme. So the organism basically produce your urease enzyme which will split down the urea, and that is your urease production test. So moving on to the principle behind your urease production test. So the principle behind the urease production test is as follows. So urea is produced or the product of decarboxylation of amino acid. So if I write down, this is your amino acid. So this amino acid will undergo decarboxylation process, and this will ultimately produce your urea. Okay. Now the hydrolysis of urea, so this urea which is being produced is further being hydrolyzed. So hydrolyzed to produce your ammonia. So ammonia and your carbon dioxide. Okay. So the formation of ammonia. So when we are having a lot of ammonia in your culture medium, the ammonia will alkalinize the medium. So it will make your medium alkaline. And the pH shift. So when there is a change to its alkaline medium, there will be a pH shift, and that particular pH shift is detected by the color change of phenol red. So you have a indicator phenol red. So this is your indicator phenol red, and this phenol red will actually convert this particular color into your from light orange. So it will convert the color from light orange to your magenta or your pink. So magenta or your pink, and that will be done within 24 hours. So you have 24 hours within which this phenol red will change its color from light orange to magenta. So that is the indication of your rapid urease positive organism turning the entire medium from your orange to your pink within 24 hours. Now there are some weak positive organisms, may take several days. So these are the strong positive organisms. But there are certain weak positive organism also. So what does this weak positive organism do? They will also do the same thing, but they will take a several days. So they will take several days to bring about this particular result. Whereas, the negative organism produce no color or yellow as a result of acid production. So the negative organism, so it is if it have a negative organism, then the color will be changed from the light orange to your yellow. So light orange to your yellow. So it is changed to your yellow, and that is the result of your acid production. So that is the result of your acid production. So now I think you have come to understand that this acid production will change the color to yellow, whereas the alkaline production will change into a magenta color. Okay, so now I will write down the formulae of the overall reaction. So the reaction which is going on, that is the basic principle of your urease production test. So I will write down the equation. So the equation is NH2O twice CO, that is your urea, plus twice the molecule of your water in presence of your urease will produce carbon dioxide, water molecule, and your 2 molecules of your ammonia. So carbon dioxide, water molecule, and ammonia, and this ammonia will make your particular test positive because of the change in the color of the medium. Okay. So this test is sometimes used, right, in your urea breath test. Okay, I will give you an example. Means I will just relate it to an example. Like that there are certain organisms which you have also studied in your medical microbiology, Helicobacter pylori. So Helicobacter pylori, okay, it's a Helicobacter diagnostic test for the diagnosis of Helicobacter pylori. It's a rapid, cheap, and simple test that detects the presence of urease in or on the gastric mucosa. So you just find out urease in your gastric mucosa. So you find out in the gastric mucosa, and it is also known as your Campylobacter-like organism test. And this test will tell you that whether you have your Helicobacter pylori or not. The urease produced by this particular organism, Helicobacter pylori, will hydrolyze the urea, okay, which is being given to the patient, to your ammonia, and you will be able to smell the presence of ammonia in his or her breath, which is also called as your breath test, urea breath test. Okay, and that which will rise the pH of the medium and change the color of the specimen from yellow to red. So that is another way by which it can be distinguished or diagnosed. Okay, so this is also a part of your urea breath test, and urea breath test is being done as a non-invasive test for those who are suffering from your from your peptic ulcer. Okay, so next is the procedure. So that you have to know about the Christensen urea agar, and this particular urea agar, the main components are your urea, sodium chloride. So urea is 20 grams, sodium chloride is 5 gram, monopotassium phosphate. So monopotassium phosphate, peptone 1 gram, dextrose 1 gram, phenol red 0.012 gram, and if it is an agar method, if you make a plate, then you go for the agar addition, that is your 15 gram. Okay, so now I will move on to the procedure. Okay, so before we move on to the procedure, let's insert one image. So from there we can determine the procedure. So if you move on to the procedure, So this is your procedure. So if I just enlarge it, you see that these are the procedure. So first what they are doing is that transfer 3 to 4 ml of Christian Sense medium. So you are taking, this is your stock medium, and from there you have taken into the culture tube, and then you have put it into a slanting position as you are making a agar slant over it, and then you refrigerate at 4 to 8 degrees centigrade so that it's solubilized to its maximum. And then with the inoculation loop, you add the test organism, streak the test organism onto that of your culture tube, and you incubate at 35 degrees Celsius for 16 to 24 hours. And then you do the observation. You see the observation in your slant. So it is the most easiest way by which you can perform the above test. So this is about your procedure. So next we will move on to the results. So for results, again I will like to insert, of course, when I am inserting the image, I will try to acknowledge all the image which I have taken from the web page, that is from Google. So these are all the Google images which I have taken, and I will always try to acknowledge this image for preparation of my video lectures. So this is basically the result of the Urease test. So it's very easy, it's not that tough. So if it is a quick positive result, you will see after 24 hours, the color of the medium changes to pink, which I have already discussed. Why? Because of the presence of the ammonia which is being synthesized, right? And then this is a delayed positive result. A slight pink colored medium is being seen after several days, and that is your delayed positive result. And last but not the least is your negative result. And this particular negative result, you see that the medium has turned yellow. So these are the three results which you expect. So this is for the positive test. This is for the delayed positive test. So I will take a double plus over here, delayed positive test, 1 plus over here, and this is your negative test. Okay. So this is a small YouTube video. So I will also try to acknowledge the YouTube video which I am trying to display in this particular lecture, just to prepare my video slides for my virtual lab. So that is why I have taken this video. So I will always acknowledge the video which I have taken for the video preparation purpose. Okay. So let's see that whether this video opens or not. So this is actually a video which is being prepared by your you can see the name Dr. Amar. So I will acknowledge this video, and I will try to quickly show you. This is basically the students, what they will do, the blood agar, and in this there is a culture, and they have put this particular urea slants for testing of those organisms in the agar. So now you see in the first culture tube they put Proteus vulgaris, in second Klebsiella pneumoniae, in third they have incorporated Escherichia coli. So you see this is the culture of your pure culture at 24 hours. So they have taken the inoculation loop, and they have started streaking this particular culture in the culture tube. Now again the Klebsiella pneumoniae is being taken. So like this way, all the three culture tubes are actually being streaked, and then you keep it in incubator for 24 hours. And now this is Proteus vulgaris, this is your Klebsiella pneumoniae, this is your E.COLI, and control. See, the first one suggests that it is a quick positive test, total positive test, and this is a delayed positive test, because half of the agar is actually having a pink or magenta color. The whole agar has not converted into your magenta color. In E.COLI, you get to see that there is totally yellow color, the control test. So with this, I will end my virtual lab today. Thank you, students.